So this is how we conduct an experiment for the biosensor lab using crude enzyme from fruit peels. The objective, as we've seen on the screen, you want to modify the electrode with crude tissues from fruit peels. The experimental procedure. So first, all you need is to get fruit peels. You can get it from apples, you can use bananas, you can always use potatoes as well. So use a knife to peel it off, and then you start weighing them. So in this case, we weigh about 0.1 grams using a weighing scale, and then you start weighing another 1.0 gram of graphite powder. So we use a graphite powder because this is conductive material in order to modify the electrode surface. So 1.0 gram graphite powder, 0.1 gram fruit peels, and 300 microliter of a paraffin oil, because graphite powder doesn't dissolve in water, so we use oil or organic solvents. So that's 300 microliter of paraffin oil. You start pouring into the your mortar and pestle, so that you can start mixing, or what you call it, grinding. Yeah, that's how you do it. You mix them well until they are homogenized. So that's how it looks like. Now you take your empty Teflon electrode, you fill it in, you start filling in the there's a hole there until it's full, and you can start smoothing it using the glass surface, or you can use your spatula as well. So that's how you prepare a working electrode. Yeah, this is your working electrode. And now this is your potentiostat. A potentiostat is the device that you use to measure voltammetry technique. This is, as we told you before, there are three types of electrodes that you're going to use. The green one is the working one. The yellow one will be connected to a reference electrode. And then there's another one there, which is a counter electrode. We use a counter electrode to minimize all the noises from the experiments. You just start connecting them. Then you set up the setting on the screen of the potentiostat. You start running from within your selected potential window. As the experiment goes by, you will get results. You get signals. You can also name. You can start naming whatever response you get. So in this case, you start running from about minus 0.5 volt. I think the scan rate was 50 millivolt per second, and there you go, the oxidation peak, and then it goes back, and then you go and you see a reduction peak. This is a reversible reaction. This is a good example of a good voltammetric technique response. We call this voltammogram. The picture or the data that we get from this technique is voltammogram. So as I told you, we start scanning from a negative potential. In this case, from minus 1.0 volts, we go to from left hand side to the right hand side. So we gradually, as we scan, we will see we should be seeing an oxidation peak. There you go. That's the oxidation peak that you get from this reaction. Oxidation peaks all normally appears at a more positive potential, and once the scanning reaches the plus 1.5 volts, it goes back. That is why we call it a cyclic, because it's a full cycle. It goes back to where it starts. So now everything that we see is a signal from reduction process of the molecule. In this case, we are talking about quinone, right? A phenolic compound. So now we are looking at the formation of a reduction peak. That we start looking at a reduction peak. Reduction peak always appears at more negative potential. Then the scanning stops at where it starts.